Thank you very much, and thank you very much for inviting and all of you for being here. So patients with REM sleep behavior disorder are characterized by the loss of the normal atonia that will occur during REM sleep. And as you can see from the example in the video, they act out their dreams. This means that they have a very restless sleep, they have sleep disturbance, and they can hurt themselves or their bed partner. But most importantly, they have a high risk for Parkinson's disease or a related disorder. And in fact, 75% will develop Parkinson's disease or a related disorder within 12 years. This risk factor, unlike other factors that are shared with other neurodegenerative diseases, is highly specific for Parkinsonian disorder. And therefore, this is a good opportunity to study the prodromal phase of Parkinson's disease. Since we know the phases of Parkinson's disease, we know that it goes through the Braak stages, we know that this disease starts from the brainstem through the basal ganglia and then it spreads to the rest of the brain. So the question we asked in this study was, can we track dopaminergic neurodegeneration in prodromal Parkinson's disease? And to do this, we focused on two structures of interest, the locus coeruleus-subcoeruleus complex, which controls REM sleep, and the substantia nigra, which is more related to the motor features of Parkinsonism. And as you can see, these structures in histology appear dark, and this is because they contain dopaminergic neurons, which contain neuromelanin, which is a pigment that is dark in histology, but due to its magnetic properties can be seen in on MRI using neuromelanin sensitive MRI. And as you can see, in this case, it appears it appears bright on MRI, sorry. This is a technique that has been around for pretty much more than 10 years. And actually, it has been shown that in Parkinson's disease, measures derived from neuromelanin sensitive MRI can distinguish patient from controls, and they are correlated with the severity of Parkinson measured with UPDRS. There are also two studies in RBDs showing that in the locus coeruleus we have a decreased neuromelanin in RBDs compared to controls, and also a decreased volume of the substantia nigra. However, all these studies to be able to measure the bright voxels on MRI, they use manual selection of either the search region or a manual placement of the reference region used to detect the bright voxels. And this has been done for each and every subject. So the first aim of our study was to develop an automated and objective way to quantify to detect these bright voxels in the key structures. And the second key question is that even if there are two studies in RBDs, they show the difference between patients and controls. But some key questions remain to be answered within the RBDs. So who will convert, when they will convert, and to what type of Parkinsonian disease? And actually, before being able to answer these main questions, we still need to find a link to basal ganglia denervation and to the Parkinsonian risk. So to do this, we enrolled subjects from the Oxford Parkinson's Disease Center discovery cohort, in particular 37 RBDs that underwent MRI, nuclear imaging with that SPECT CT to be able to link to the basal ganglia denervation, and clinical evaluation, which included the Movement Disorder Society probability risk score for Parkinsonian disease. And we also had 25 healthy controls matched with the patients that underwent MRI. Regarding MRI, the neuromelanin sensitive MRI is a T1-weighted scan with a magnetization transfer pulse. We have a reduced field of view, so we need a full brain MPRH T1 to be able to help the registration. We first developed an automated method to extract measures from a neuromelanin-sensitive MRI, and then we compared these measures in terms of their ability to differentiate RBDs versus controls. So first of all, replicating previous findings, differentiate the RBDs between those who had a normal or abnormal DAT scan, as reported by a neuroradiologist, and to correlate those measures with the clinical risk for Parkinsonian disease. Regarding the automated method, we first of all built a study-specific template of neuromelanin MRI using a mix of patients and controls, both for the substantia nigra and the locus coeruleus. We then placed the search regions, red and blue, and the reference regions on the template in MNI space. And then we registered, we transferred back the ROIs into the individual scans, and then extracted the volume of the substantia nigra and the locus coeruleus based on the mean and standard deviation intensity within the reference region, as reported in the literature. And moreover, we also extracted other measures that have been used in the literature before related to the intensity within the volume to be able to see how they compare to each other. And what we found, first of all, we found what other people found, which is good, a difference between patients and controls in both the volume in the substantia nigra and in the locus coeruleus. And in particular, we can see that these two clusters are not completely separated, which to us it was reassuring because let's remember that these RBD patients, some of them will develop Parkinson's soon, some of them will develop it later, some of them will not develop it at all. So there is some sort of overlap with controls. Regarding the nuclear imaging, we had 20 RBDs with normal DAT SPECT CT and 17 with abnormal DAT scan. 11 of which they were abnormal because of asymmetric uptake, because since Parkinson's is an asymmetrical disorder, asymmetry of the uptake is considered an abnormality, and 6 of them had generalized signal loss. So between those two groups, if we go back to the MRI, we found that not the volume, but the peak intensity in the substantia nigra was different between subjects who have a normal versus abnormal DAT scan. And also, if we calculate the asymmetry between the asymmetry within the nuclear imaging and the asymmetry within the substantia nigra, we found a correlation, in particular, between the asymmetry in the putamen uptake on the DAT scan and the asymmetry in the substantia nigra volume. Regarding the correlation with the clinical risk, we found a correlation with the volumes, both in the substantia nigra and in the locus coeruleus with the clinical risk. So to conclude, we developed a fully automated method to extract measures that can be linked to prodromal Parkinson's from neuromelanin-sensitive MRI. These measures are related to dopaminergic deficit that we measured with DAT SPECT CT and to the clinical risk using the clinical risk scale. Interestingly, different measures may be more sensitive to different aspects of the disease process. And obviously, the significance of the finding will be ultimately be determined by the ability of these measures to predict the conversion at follow-up, which is already planned in our cohort. And obviously, another key significance will be to be able to track the progression and the changes of this over time. And with this, I would like to thank all my collaborators, in particular Tom Barber and Claire Mackay, all of you for your attention. And if you have any question, I can take them now or at the poster session.